So after cells of epidermis, we will be discussing the layers of epidermis. So next heading is now in the layers of epidermis, I already told you that the most superficial layer is stratum corneum, the deepest layer is stratum basale, in between there is lucidum, which is the condensed portion of stratum corneum, then G comes first, so granulosum, then comes S, so spinosum, and finally the basal layer. Okay. Now remember one point, that they develop from below upwards. Okay. So when this epidermis develops, these layers, they develop from below upwards. So which is the first one to be formed? Stratum basale. And which is the last one to be formed? Stratum corneum, isn't it? And that is the reason why they ask you this question, that in a very low birth weight baby or in a preterm baby, which of the following layer of epidermis is deficient? And the answer is stratum corneum. So in a very low birth weight baby or preterm baby, this layer is deficient. Okay. So let us discuss the layer which is formed first. So we will begin our discussion with stratum basale. So highly mitotically active keratinocytes with housekeeping organelles. So highly mitotically active keratinocytes with housekeeping organelles are seen. Okay. So they are highly mitotically active, and what mitotically active cells do? They proliferate. Okay, so because they are proliferating all the time, they require all the organelles which are important for that, the mitosis, and that's why these cells, they have all the housekeeping organelles like Golgi bodies, mitochondria, so they are rich in that. So let us consider that this is stratum basale. These are the basal columnar keratinocytes. And because they are highly mitotically active, what they are doing, they are proliferating. So when they proliferate, they form other cells, then other cells, and these cells, they gradually keep on moving upwards, and soon they reach the topmost layer, that is stratum corneum. Okay. Now they cross stratum corneum also. Soon they reach the topmost layer of stratum corneum and they start sloughing off from here. So finally, they are sloughed off in the environment, isn't it? So what is the destiny of these cells? These cells, they start proliferating from stratum basale, they keep on moving upwards, they cross all the layers, they reach stratum corneum, and finally from the topmost layer of stratum corneum, they slough off in the environment. Okay, so this total time which is required by these cells to move from stratum basale and finally shed off in the environment, this total time is known as epidermal turnover time, or skin doubling time. So the total time which is required by these cells present in stratum basale to finally shed off in the environment is known as epidermal turnover time and skin doubling time. Now with the help of various experiments, we tried to calculate this time, and we found that these cells which are present in stratum basale, they require around 14 days to move from stratum basale and reach the lowermost portion of stratum corneum. Then they require another 14 days to cross stratum corneum, and once they cross stratum corneum and reach the topmost portion, this sloughing off is a very gradual process, and this sloughing off requires another 28 days. So this total time becomes 56 days. So first 14 days are required to move from stratum basale to lower part of stratum corneum, another 14 days to cross stratum corneum, and then 28 days to slough off. So total time becomes 56 days. So if they ask you in exam that epidermal turnover time is or skin doubling time is, answer is 56 days. Now this point is slightly controversial. Why? Because there are so many different books and they give so many different answers. Like Arvind Arora Guide and Davidson, in both it is written epidermal turnover time is 28 days. Okay. Now, if you read specific textbooks of dermatology like Nina Khanna textbook of dermatology, which is very famous in UG graduates, so there it is written epidermal turnover time is 60 days. Okay. In my book, so book by Manish Soni, CBS publication, there I have written 56 days, and here also I am telling 56 days. Now, if you read Rook's textbook of dermatology, there it is written that epidermal turnover time is 52 to 75 days. So in different books and different guides, you will get different numbers, okay? But I would suggest you to remember this, that is 52 to 75 days. This also falls in this range, so this is correct. This also falls in this range, so this is correct, but this is not correct. Okay, so remember that epidermal turnover time is 52 to 75 days. If specifically asked, you can mark 56 days. The confusion arises because in one of the guides this question is there that epidermal turnover time is, and the options are 1 week, 2 week, 3 week, 4 week. Now, because these are the only 4 options, you need to choose one, and the answer given is 4 weeks, so that is correct. But if you have options like 2 week, 4 weeks, 6 weeks, 8 weeks, then you should mark the one which falls in this range. Okay, that is 52 to 75 days. Okay, so remember this range. If it's specifically asked, then it is 56 days. Okay, so epidermal turnover time is 56 days, or range is 52 to 75 days. Okay, but this time is drastically reduced in one of the condition, and that condition is psoriasis. In psoriasis, epidermal turnover time becomes 4 days. So you can imagine what is happening in psoriasis. In psoriatic skin, in the epidermis, the cells are very quickly moving upwards and they are very quickly sloughing off within a period of 4 days. Okay, so the thing which happens in 52 to 75 days is happening in 4 days in cases of psoriasis. So you can imagine what is happening. The sloughing off is so quick that so much of cells are sloughing off that they become visible, and they are visible in the form of scaling. So now you know that why psoriatic lesion have so much of scaling. Why? Because there is a lot of sloughing off, epidermal turnover time is reduced. Okay, let us move on to the next layer, which is stratum spinosum. Stratum spinosum, this is also known as prickle cell layer. Stratum basale is also known as stratum germinativum. Why? Because it is germinating from there, it is proliferating from there. This is prickle cell layer. Why prickle? Because when you will see this layer, you will find multiple pricks or spines over the cells, like you can see in this image. So this is stratum spinosum. These are the keratinocytes which are present here, and if you look at their surface closely, you will find these spine-like processes. Isn't it? They are visible. So what are these spine-like processes? So if this is a keratinocyte and this is another keratinocyte, both these are attached together via desmosomes. Both these are attached together via desmosomes. So desmosomes are the intercellular structures which help in attachment of 2 keratinocytes. Okay, so this is an intercellular structure which help in attachment of 2 keratinocytes, and under microscope they look like spines or pricks. So because they are prominently present in this layer, that is stratum spinosum, you can see lot of these cells having spines over them. That's why this layer is known as spinosum. So this is so simple to remember. Spinosum, lot of spines are seen. So you can write down first point, desmosomes are prominently seen here. And this was the question which was asked in NEET exam, that desmosomes are prominently seen in which of the following layer of epidermis, and one of the option was stratum spinosum. So remember, they can be seen in other layers also, but prominently they are seen in stratum spinosum. So that's why this is known as spinosum or prickle cell layer. Now these desmosomes are important, so let us write down few more points about them. Desmosomes are made up of desmosomal proteins. So desmosomes, they are made up of desmosomal proteins, and these proteins, they require calcium for their function. And what is their function? Attachment of two cells. So that's why they are known as calcium dependent adhesion molecules. So desmosomal proteins, also known as calcium dependent adhesion molecules. Like other proteins, these also belong to following protein gene families like cadherin. Already question have been asked, cadherin, plaquin, armadillo, and these are their examples. So they belong to, so these desmosomal proteins, they belong to cadherin, plaquin or armadillo gene family, and their examples are desmoglin, in short written as DSG, desmoplakin, DP, desmocolin, DSC, placoglobin, PG, placofilin, PP. So these are various examples of desmosomal proteins like desmoglin, DSG, desmoplakin, DP, desmocolin, DSG, placoglobin, PG and placofilin, PP. Among them, this one is very important. Why this one is so important? Because this is the desmosomal protein which is involved in pemphigus. So all of you might have read about this blistering condition, that is pemphigus. Pemphigus is very important. Already questions have been asked, and sometimes they ask you that which of the following desmosomal protein is involved in pemphigus, and the answer is desmoglin. So in pemphigus, which protein is involved? Desmoglin. So now you know, just by knowing this simple point, you can answer various questions which are asked on pemphigus. Like they might ask you that in pemphigus, which type of blister is seen? Intraepidermal or subepidermal? So how to approach to this question? Now we know that pemphigus, which protein is involved? Desmoglin, and desmoglin is present in desmosomes, and desmosome help in attachment of keratinocytes, and keratinocytes are present where? In epidermis. So if the separation will occur, it will also occur within epidermis, and due to which if blister is formed, that will be also present where? Within epidermis. So it will be called intraepidermal, isn't it? So not only desmoglin, all these proteins are present in desmosomes, that is within epidermis. So if any of these protein will become defective, it will lead to which type of disorder? Intra or subepidermal? Intraepidermal. So if any of these proteins becomes defective, it leads to intraepidermal disorder. Okay. So sometimes these questions are asked that which of the following condition is intraepidermal or subepidermal, and you can very easily answer these questions if you just remember the defect. If you know that what is defective in that condition, and if the defect is among these, it is going to be intraepidermal, isn't it? So later on we will be discussing various intraepidermal and subepidermal disorders, but for now just remember that defect in desmosomal proteins lead to intraepidermal disorder, and one such example is pemphigus. Now you need to remember these proteins because later on we will be discussing them in intraepidermal blistering disorders. Okay, so don't forget. Okay, so let us revise again. Desmoglin, desmocolin, desmoplakin, placoglobin and placofilin. All these are desmosomal proteins, also known as calcium dependent adhesion molecules. Next is stratum granulosum, which is also known as granular cell layer. Also known as granular cell layer. Now this is again very obvious from the name itself, granular cell layer. So it might be having various granules, isn't it? So two important granules you need to remember. The first one is keratohyalin hyaline granules and membrane coating granules. Keratohyaline granules and membrane coating granules. Membrane coating granules are also known as Odland bodies. Now this keratohyaline granule synthesize pre-filaggrin. Okay. Now filaggrin is a protein and this pre term is added to it because this is a precursor molecule for filaggrin. So pre here is for precursor. Now they might ask you that what is the function of this protein? So remember, it helps in filament aggregation. So this is filament aggregating protein. This function is so simple to remember because the function is hidden in its name itself. It is filament aggregating protein, filaggrin. So filament aggregating protein. So this is what? This is filament aggregating protein, filaggrin. Similarly, this membrane coating granule, which is also known as Odland bodies, synthesize lipid. And what this lipid do? This lipid forms a protective barrier. So what is the function? It forms a physical or a protective barrier. Now sometimes what happens, there is defect. Like if there is deficiency of filaggrin or if there is deficiency of lipid, it might lead to certain disorder. So the disorder which occurs due to deficiency of filaggrin is ichthyosis vulgaris, and the disorder due to deficiency of lipid is steatotic, which is also known as winter eczema, also known as eczema crackle. So ichthyosis vulgaris is due to deficiency of filaggrin. Steatotic or winter eczema, also known as eczema crackle, is due to deficiency of lipid. So these are two important disorders which might arise when there is defect in these granules or this protein which is being synthesized by these granules. Now we will be discussing these disorders later, but for now just remember a few points about them. Ichthyo means what? Ichthyo means fish. So fish like scaling is seen. And do you know what is the meaning of this term vulgaris? Vulgaris means most common. So wherever you find this term vulgaris written, you can very easily conclude that that is the most common disorder. Okay, like ichthyosis vulgaris is the most common type of ichthyosis. Acne vulgaris is most common type of acne. Lupus vulgaris is most common type of cutaneous tuberculosis. Verruca vulgaris is most common type of warts, commonly known as common warts. Then vitiligo vulgaris is most common type of vitiligo. Few years back in DNB question was asked which is the most common type of psoriasis. It is psoriasis vulgaris. So remember, wherever you find this vulgaris term, that means that is the most common variant of that disease. So ichthyosis vulgaris is the most common type of ichthyosis. Because it is ichthyosis, here fish like scaling is seen, and this is due to deficiency of filaggrin. Okay. Now steatotic eczema or winter eczema. The aggravating factor or the trigger here is cold or winters. So this eczema generally occurs in winters due to cold. Okay, once this question was asked that steatotic eczema is endogenous type of eczema or exogenous type of eczema. This is very obvious from the name itself. Endogenous, that is the cause is inside, or exogenous, the cause is outside. Okay, so because it is winter eczema, everyone thought that it is due to cold, and cold is outside the body, so it is exogenous. But remember that this is endogenous type of eczema. Why it is endogenous? Because just now I told you it is due to deficiency of lipid. So though cold is a aggravating factor, it is a triggering factor, because when there is deficiency of lipid, the skin becomes more dry and cold. But remember that the cause is actually deficiency of lipid. So this is endogenous type of eczema. Okay, so remember these 2 points. Now sometimes question is asked that filaggrin is present in. So I already told you that pre-filaggrin is present where? In keratohyalin granules, is being synthesized by keratohyalin granules, where? In stratum granulosum. So pre-filaggrin is present in stratum granulosum, and this is a precursor molecule. Okay, so this precursor molecule, when get matured, by the time these cells move above, and which is the layer present above stratum granulosum? That is in stratum corneum. Okay, so in stratum corneum, so except for palm and sole where stratum lucidum is present, which is the layer above stratum granulosum, it is stratum corneum. So filaggrin is present in stratum corneum, pre-filaggrin is present in stratum granulosum. Similarly, Ordland bodies. Ordland bodies also I have told you they are present in stratum granulosum, but if stratum granulosum is not there in the option, you can choose stratum. So answer will be stratum granulosum, but if it is not there, you can choose upper part of stratum spinosum. Why? Because let us consider that this is stratum spinosum, this is stratum granulosum. Okay, so what happens that these Ordland bodies, they start forming here in the upper part of stratum spinosum. Gradually they move upwards and they reach stratum granulosum. So if stratum granulosum is not there, choose upper part of stratum spinosum. Now this is the site where they are present in skin, but these Ordland bodies are also present in lungs. In skin they synthesize lipid, in lungs they synthesize surfactant. In type 2 alveolar cells, they form surfactant. So remember, Ordland bodies, they are present in skin, in stratum granulosum. If it is not there, upper part of stratum spinosum. In lungs, in type 2 alveolar cell, they form surfactant. Next is stratum lucidum. Stratum lucidum is also known as clear cell layer. It is also known as clear cell layer. Now can you tell me why it is known as clear cell layer? Clear, because the cells which are present here, they are clear. What does that mean? The cells present here are translucent. Now why these cells are translucent? Because of presence of refractile granules of elidin. So this is one important protein which is present in these cells, that is elidin protein. So these cells, they are filled up of elidin protein, and this is refractile in nature, and because it is refractile, these cells, they appear translucent or they appear clear, and that's why this is known as clear cell layer. Okay, so stratum lucidum is clear cell layer. It is present in palm and sole. So remember that this layer is present in palm and sole, and that's why the epidermis over palm and sole is thickest. Why? Because there is one extra layer which is present. Okay, so the epidermis over palm and sole is thickest because of presence of one extra layer, that is stratum lucidum. Now because this epidermis over palm and sole is thickest, when the lesions appear over palm and sole, sometimes they appear deep seated because of this thick covering. Okay, and such lesions can be seen in a condition which is known as pompholex. So let us write down one point about pompholex here itself. Pompholex. It is a type of eczema. It is a type of eczema where deep-seated clear fluid-filled vesicles. See, vesicles are very small fluid-filled lesions. They are generally less than 0.5 centimeter in size. So deep-seated clear fluid-filled vesicles are seen over palm and sole, which are better felt than seen. So they are better felt than seen. So if you palpate them, they are very easily palpated, okay, but if you see them, because they are having clear fluid filled, so they are not very clearly visible, but if you palpate them, you will get a characteristic feel, so which are better felt than seen, giving sago grain or tapioca feel. So these are the points which you need to remember about Pompholex. Okay, so once this question was asked on Pompholex, so you can remember a few points about Pompholex. First, it is a type of eczema. Common site is palm and sole. Over palm and sole, what you will get? You will get these deep-seated vesicles, fluid-filled vesicles, and these vesicles, they are better filled. These lesions, they are better felt than seen. And when you feel them, which feel you will get? Sago grain or tapioca feel, sabudane jaisi feel. Okay. Next is stratum corneum. Stratum corneum. Now, stratum corneum is the topmost layer, and in this topmost layer, if you see the cells, all of them are dead keratinocytes. Dead keratinocytes. So, stratum corneum is made up of dead keratinocytes. Why we call them dead? Because they lack nucleus. So, nucleus is absent. Okay, so because nucleus is absent, this layer don't have any mitotic or metabolic activity. The cells which are present here are generally flat and they are dehydrated. So these are the characteristics of the keratinocytes which are present here. They are very flat, they are dehydrated, they don't have nucleus or mitotic activity, and all of them are, because they don't have nucleus, they are dead keratinocytes. Okay. So these are the characteristics which are seen in these cells, and gradually as we move upwards from stratum basale, you will find these changes. Like if this is stratum basale and this is stratum corneum, if you keep moving upwards, you will find that gradually all cells, they become flat. Okay, so all cells gradually they are becoming flat, and because they are becoming flat, the overall surface area increases, so they appear large. Okay, so the size is increasing. So as the cells move upwards, they become flat, they keep on increasing in size, and because they are moving outwards, their moisture is lost, so they become dehydrated and they lose their nucleus. So nucleus is lost. So in the lower layer, nucleus is very prominent, but in the upper layers, nucleus is lost. And because nucleus is lost, the metabolic and mitotic activity is also lost. So these are the changes which occur as we move upwards. Like you can see here in the lower layers, so like basal layer, spinosum, granulosum, you can see nucleus. So nucleus is very prominent in basal layer and it can be seen in other layers also, but in the upper layer, like you can see lucidum and corneum. So till lucidum you might find nucleus, but in corneum you won't find nucleus. So that's why these are dead cells. So these are dead cells in stratum corneum. These are dead cells. They don't have nucleus. They are sloughing off. And now you know that why when we move from stratum basale to stratum corneum, we require only 14 days. And to cross just single layer, you again require the same time, that is 14 days. Why? Because the nucleus is absent. These cells are not proliferating. They are simply being pushed upwards, and that's why they require more time. And once they reach the topmost layer, again to slough off, they require 28 days. Okay, so that's why the time is increasing, because the mitotic activity is decreasing. So this completes the layers of epidermis as well. Now you can write down one important point here about desmosomes. See, I already told you that desmosomes have desmosomal proteins, and I told you the names of those desmosomal proteins as well. Now, if you closely examine them under electron microscope, you can see the various proteins and their arrangement, and they are arranged something like this. So let us consider that these are two keratinocytes. So these are two keratinocytes and they are attached together via desmosomes, and desmosomes have desmosomal proteins. So these desmosomal proteins are arranged something like this. On the inner side, other desmosomal proteins are present. So let us label them. You will find desmoglin and desmocolin. So you will find alternate desmoglin, desmocolin, desmoglin, desmocolin, which are attached to placoglobin and placophilin, which is further attached to desmoplakin, and which is further attached to keratin filaments, also known as intermediate filaments. Okay, so this is the arrangement of desmosomal proteins. On either side you will find same, that is desmoglin, desmocolin, then placoglobin, placophilin, which is further attached to desmoplakin, which is further attached to keratin filament. So like you can see here, these are keratinocytes, all these are keratinocytes, and these are the filaments, and in between they are attached via these desmosomes. So these are desmosomes. So you can again see these are the intermediate filaments, that is the keratin filaments, and they are attached together via desmosomes. And desmosomes, I told you, they belong to which gene family? Cadherin gene family. It will become more clear when we further magnify and see that this is keratinocyte, this is keratinocytes. They are attached together via alternate desmoglin, desmocolin, which is further attached to placoglobin, placophilin, which is further attached to desmoplakin, and this is further attached to intermediate filaments or keratin filaments. So these are intermediate filaments or keratin filaments. Okay. Now you can see that there are two junctions, that is, this is the first junction where all the proteins are attached to each other. And similarly, this is the second junction, like here the placoglobin, placophilin is attached to desmoglin and desmocolin. Similarly, this is the second junction where desmoplakin is attached to intermediate filament. So these two junctions, when you see them under electron microscope, these junctions, they appear electron dense. So this is the outer electron dense part and this is the inner electron dense part. Okay, so these are the junctions and these are the arrangement of desmosomal proteins within a desmosome. Okay.